All right, welcome everyone. Welcome YouTube and everyone on who may be on Zoom or in the room. So today is our second Water, Wetlands, and Watershed Seminar of 2025. And today we're lucky enough to have with us from the Keys. You're calling us from, what key are you on, Shelley?
Key West.
All the way down in Key West. Raise your hand in the room if you've been to Key West. Oh, look at that. I don't know if you can see in the room, but it's like most. So I have not.
Great.
There you go. You went. Oh, for the NWCA? Oh my gosh, I gotta get down there. Anyway, so thanks for, we'll come join you next time. So Shelley Kruger is an extension faculty member at the University of Florida and Florida Sea Grant agent. She's been in the Keys since 2013. For those of you who don't know, Florida Sea Grant is a UF-based program that supports research, teaching, and extension to conserve coastal resources. Shelley has her BS from Georgia Tech and an MS from Savannah State University in marine sciences, and she's also a PhD student at the UF School of Forest, Fisheries, and Geomatic Sciences in Fisheries and Aquatic Sciences. So Shelley, you're a full-time student and a full-time extension agent?
Correct.
Okay, so if you think your low schedule is hard, right, then you look at Ms. Kruger here and see the soon-to-be Dr. Kruger is gonna really show us what you can do. So all right, in 2019, Shelley wrote the sponge restoration module for the IFAS Florida Master Naturalist Program. And as an extension agent, she provides science-based education for Florida's coast on a broad range of topics, including sponges, which we'll hear about today, water quality. I don't know if we'll hear about sargassum or the giant blob that's been threatening Florida over the past few years, as well as stony coral tissue loss. So Shelley, the floor is yours. Thank you for joining us.
Thank you so much. When most people think of the Florida Keys, they think of Florida's coral reef, but in fact, the majority of the Florida Keys is dominated by seagrasses and hard-bottom habitats that support over 60 species of sponges in the nearshore waters. Unfortunately, since at least 1991, there have been a series of sponge die-offs in Florida Bay and the lower Keys that have decimated these active ecosystems. And I'd like to start by asking the class if anyone would like to venture a guess on a definition of restoration aquaculture.
All right, Shelley, we'll give them a minute because sometimes it takes a second to get the gears turning. So
Sure.
restoration aquaculture. Has anyone heard of aquaculture? All right, so how about an aquaculture definition? Come on, it can't be wrong. There's no like, it's just pass fail. Come on, what we got? Yeah, go ahead.
Like the growing or controlled controlling of underwater species and plant species. I don't know if it includes animal species involved in this, but maybe things like clams and things like that as well, but something loosely like that.
Okay, so aquaculture, we got like growing underwater species for harvest.
Yeah.
Okay. How about restoration? Anyone want to give a restoration?
Well, if I had to assume, it would be the restoration of where aquaculture has been impacted in some way, shape, or form, like trying to restore it to its natural state that it was in prior to being disturbed.
So Shelley, I don't know if you could hear that, but that was like restoring aquaculture to a previous state. All right. Let's do one
Absolutely.
more.
Oh yeah, one more.
No, please go ahead, Shelley. Yeah, continue.
Okay. Yeah, so absolutely. Restoration aquaculture is just a combination of those terms. And the Florida Code definition is restoration aquaculture is the cultivation of plants and animals to increase declining wild populations and improve the health of coastal ecosystems. And so sponge restoration is a component of this. It's a nature-based solution that improves water quality, biodiversity, and essential fish habitat. And in essence, restoration aquaculture sponges can help alleviate the long-term ecological damage caused by, oh, sorry, my can't read my slides with all this stuff on it. Let me just move that over real quick. To alleviate the long-term ecological damage and improve water quality. Okay. So I've been working with the Florida Fish and Wildlife Conservation Commission on sponge restoration for at least the past 10 years. For those of you who are unfamiliar with sponge restoration, here is a photo of sponge propagules inside a FWC permitted nursery near Marathon, Florida. And many of you are probably familiar with coral fragmentation. And so sponge restoration manipulates their asexual reproduction in the same manner that they do for coral fragging. So we collect a fist-sized cutting from adult donor sponges, zip tie them to bricks, and place them inside these FWC nurseries where they can cure and heal. And then at some time in the future, they're moved to die-off areas. And so these propagules right here were collected from adult donor sponges, and then they are in this monoculture at the moment while they're healing, and then they will be moved to other areas with other sponge species enhance that biodiversity. And so these are base sponge propagules at time 0. This is a photo of a base sponge propagule 2 years later. You can see that the base sponge has reformed its morphology. You can still see the brick and the zip tie underneath it. And these are base sponge propagules 4 years later. And that red arrow is pointing to there's already recruitment of other sponge species at the base of these base sponges. The sponge has literally completely covered the brick, and it would take a crowbar to remove these from the benthos. And really, they look identical to the neighboring, adult sponges. And here are some other species that we use in sponge restoration aquaculture. On the top, A is a base sponge. You can see how it's starting to reform that basket-shaped morphology. B, you can see how they very quickly overgrow the zip ties and begin to reform their traditional shapes. We have a funny one down on the right. We actually call that elf sponge. It's Clyonivarians, and it's very common weedy sponge. It grows in a bunch of different morphologies, hence the variants in the species name. But for those of you who remember the sitcom in the late 80s about an alien who comes and live with the family, his name was Elf. And this one in particularly really grew to look like an elf sponge. And then here's an example of some other sponges that we use. Along the top is a loggerhead sponge, another base sponge, brown branching. Along the bottom, golf ball sponge, yellow sponge, and mountain or glove sponge. And so now we are going to transition into talking more about water quality and ecosystem services. So as I mentioned earlier, this is a map of the benthic habitats of the Florida Keys. A large portion is seagrasses and hard bottom habitat that is prime habitat for nearshore sponge species. And these are highly productive ecosystems and are especially important nursery grounds for many species of recreationally and commercially important fish and invertebrates. So sponges are filter feeders. And they're really feeding on the smallest fraction of phytoplankton called picoplankton. And each sponge species actually kind of has its own niche of the size of this picoplankton it's filtering. They are also filtering, and by filtering, they're feeding. So they're feeding on picoplankton, bacteria, and viruses. They also provide essential fish habitat for juvenile spiny lobster and stone crab, bonefish, which is a very important recreational catch and release species in the Florida Keys. And just these 3 species alone contribute over $150 million to the local economy every year. And so sponges have, a lot of them have these internal canal chambers that support an abundance of animals, including the only known eusocial marine species. On the left here is a snapping shrimp. These are actually very small. Even the largest ones are only about the size of my pinky nail. And also Dr. Mark Butler and Dr. Jack Butler have done some really interesting studies on the the snap, crackle, and pop that these shrimp make. A lot of times when you're underwater in the near shore waters, you may hear these sounds, and they're not parrotfish. It's more of a snapping noise, literally. That's why they're called snapping shrimp. And they found that these are really important acoustic signals for larval fish and invertebrates to know that this is a good habitat for settlement. So they're actually really important in these auditory clues for attracting other animals to the area. And they have found that species abundance and richness is higher in sponge areas with sponge habitat versus barren sediments. And so I wanted to ask the class once again, can anyone name any land-based examples of a eusocial animal? It's kind of a unique word. So
Does anyone know what a eusocial animal is? Does anyone know what the prefix EU means? Shelley, I'm just riffing here. How about another word that starts with EU?
Eutrophication?
Which
I'm not
means
sure if that would
of
be the root of
growth.
this.
Anyone in the Gator band? What's that instrument from orchestra? The euphonium with the good sound. Right?
So
Eulogy is some good words about
yeah,
someone.
so it propagates more like biodiversity, like around it, like kind of like this.
Close. So the social part, you know what that means, right? And then the U is sort of like good or like helpful or yeah, right? So these eusocial animals, are we eusocial? Shelley, are humans eusocial? Or is that a philosophy question?
So I'm gonna skip to the answer just for the sake of time. But so an example of eusocial insects on land are bees, ants, and termites, where there's a queen who does the reproduction, and then there's a division of labor with workers and different types of that. So these snapping shrimp are, as far as I know, the only example of eusocial animals in the marine environment. And most of these snapping shrimp probably never leave the internal canals of the sponge during their entire life cycle. And most of them are also all related to that single individual who's the reproductive female. So just an interesting tidbit for trivia night. So because sponges, they can't sting, they can't bite, they can't pinch, they can't run away, they've developed a lot of chemical defenses. And this may be within the tissues themselves or within a whole host of endosymbionts that they host within their tissues. And so for this reason, they're a source of many human drugs for cancer and viruses. And according to Shayla et al., more novel bioactive compounds are obtained from members of this phylum each year than any other marine taxon. And the photo on the right is a local sponge, Tectotethia crypta. And this sponge was one of the very first marine animals to provide human drugs for the herpes virus, leukemia, HIV, and it has a lot of other anti-tumor capabilities. So for the same reason that sponges have these highly developed chemical defenses, they have very few predators, and the predators that they do have tend to be very specialized, like hawksbill turtles that feed almost exclusively on sponges, starfish, butterfly fish, angelfish, trunkfish, cowfish, and nudibranchs. So they have very few actual predators in the wild, other than these specialized ones. So unfortunately, since at least 1991, there have been a series of harmful algal blooms that have decimated sponge populations in Florida Bay. In the summer of 2023, we had very high ocean temperatures, record highs, and for a very long duration. And so we recorded a massive sponge die-off in the lower Keys, and we actually had mortality approaching 100% at one of our FWC permitted nurseries off Shark Key, which for those of you who've been there is around mile marker 11. But it went all the way from basically Summerlin Key out to Boca Grande. Oh, and I wanted to point out in this photo, so this is a photo in September of 2023, and you can see that the sponges are completely dead in both of these pictures. But I don't know if you can see my pointer. Down here, this coral is alive. The macroalgae looks well. We don't know the cause of this die-off, but it appeared to primarily affect only the sponges. And then we went out again last March, and the area was completely denuded of sponges. But if you hadn't known prior what a incredible sponge ground that was, you would have thought it was still very highly productive habitat, just because the macroalgae and the other benthic organisms were seemingly unimpacted. So while we don't know the reason for the 2023 die-off, the Florida Bay die-offs have been associated with Seneca blooms. If you see this yellow triangular area in Florida Bay just below the Everglades, since 1991, 2007, 2013, we've had a handful since then. Just entire areas where approaching 90% of the sponge species are killed, more than 25 species. And right here is a picture of a loggerhead sponge from Dr. Mark Butler when he was at Old Dominion University. And these sponges basically just disintegrate in place, and it happens very quickly. So Senecacoccus is a cyanobacteria. It is in the picoplankton. It's very tiny in terms of the fraction of the phytoplankton, its size. And it really just turns Florida Bay into this pea soup color that you can see pictured here on the right. And one thing I think is really interesting about this is that sponges feed on cyanobacteria. So this is almost an example of the prey killing the predator. And cyanobacteria are capable of producing these really sticky polysaccharides. And so they can also inhibit the zooplankton in the area. And no one really knows the mechanism of action. But once again, it seems to be primarily the sponges that are impacted by these Senecacoccus blooms. And once again, here's the photo of a loggerhead sponge before, a loggerhead sponge following the cyanobacteria bloom. This is a vase sponge before, nice and healthy, and then afterward, they just disintegrate in place. And unfortunately, this can create a trophic cascade, which can really become a vicious cycle that is difficult to return to the previous seagrass and sponge dominated ecosystems. The algal bloom can cause light attenuation and hypoxia, which can lead to seagrass die-offs. That in turn can cause, when the seagrass dies, not only are the nutrients released from the dead and decaying seagrass, but also these nutrients that have been sequestered in the sediments for decades can then be released and suspended, which can further add to the algal bloom. The sponge is traditionally helped to control the phytoplankton populations. And this in turn leads to this vicious cycle where you have a decline in spiny lobster, further declines in water quality and clarity. And ultimately, it can lead to phase shifts because it can be difficult to recover from these ongoing events. So anyway, the threats are ongoing. Unfortunately, algal blooms continue. Marine heat waves. 2023 was record-breaking temperatures, record-breaking duration. Changes in hydrology due to upland development and increased rain, drought. So if it's too fresh or too salty, poor water quality, hurricanes. During Hurricane Irma in 2017, Florida Bay actually had a reverse storm surge where the water was pulled out of Florida Bay, and a lot of these sponges remained exposed to the air hours. We don't know exactly how long, but that can lead to desiccation. And on top of the wind and wave action, really Hurricane Irma was really dramatically killed a lot of sponges. And this photo on the right, a volunteer sent me in the Upper Keys, and there were just hundreds and hundreds of dead sponges that had been dead and washed up on shore. And so this is one of the chapters from my PhD dissertation, sponge-mediated nitrogen. We measured nitrogen fluxes for glove sponge, Spongia graminia, loggerhead sponge, Spongia vesperium, and sheepswool sponge, Hippospongia lacni, in the nearshore water of the Florida Keys. Before I came to the Florida Keys, I worked with eastern oysters in coastal Georgia, which has led me to consider the similarities between oysters and sponges. And sponges are basically the functional equivalent of eastern oysters, which we do not have in the Florida Keys. And so over the past 15 years, the value of oyster reef restoration has become pretty well known. And so it is my goal for sponge restoration to be just as well appreciated. And this is why sometimes I say sponges are the new oysters. So both are efficient filter feeders. Both have been the subject of restoration efforts. There's been a lot of look into eastern oysters with nitrogen removal and oyster gardening. So we picked 3 sponge species to look at this nitrogen removal question. And so the research question was, how do 3 nearshore sponge species impact the nitrogen cycle? We picked 3 high microbial abundance sponges. As I mentioned, the mountain glove sponge, the loggerhead sponge, and the sheep's wool sponge. And the College of the Florida Keys were very generous. They let us use their outdoor flow-through mesocosms. In the blue tank on the left, I cut the sponges into tennis ball sized pieces and I left them to heal for at least 30 days. And then after the sponges healed, Dr. Ashley Smith and I placed these continuous flow chambers into the mesocosms. That's pictured on the right. And this was really ideal because they didn't have to be moved inside or anywhere else. They were in the continuous ambient temperatures that they become acclimated to. And so we conducted a static chamber incubation to look at nitrogen recycling, denitrification, and nitrogen fixation associated with these sponges. We placed 1 sponge of each species into these gas-tight chambers. And then we used syringes to remove water with replacement every 15 minutes for 2.5 hours. And what we found is that sponges are a source of dissolved inorganic nitrogen. We found that each sponge species was net nitrogen fixing. The figure on the left shows the net nitrogen fluxes in per gram wet weight of the sponges, where a negative flux is nitrogen fixation above denitrification. It's a little hard to see in here, but these are negative signs. So that's where I get the negative flux. So there were no statistically significant difference between all 3 species, but this could be due to the large variability in the sizes of the loggerhead sponges compared to the gloves and the sheep's wool. And then the figures, okay, so basically the figure on the left shows that these sponges are making nitrogen bioavailable in the water column to the surrounding species. And this isn't unexpected because if you consider historically, the Florida Keys were nutrient limited. These sponges and their microbial symbionts have likely evolved in a formerly oligotrophic environment in order to make nitrogen available in the water column. And this is a second chapter, sponge restoration aquaculture, reproductive viability following in-situ nursery propagation for nearshore spongiidae Florida Keys. So the objectives for this were to determine spawn-in months in adult donor sheep's wool sponge using histology and light microscopy, and then to survey gametogenesis following asexual propagation for 4 spongiid species at FWC permitted nurseries in the Lower Keys. So the objectives of this research are really important for natural resource management. For restoration aquaculture to be successful, ultimately these animals need to become reproductive and recruit new individuals into these ecosystems. And so right now I'm just going to talk about the sheep's wool sponge. just for your own reference, this is on the left, a female sheep's wool sponge. The circled ciliated pre-larva, these are actually fertilized internally. So the female brings in the spermatozoa from the water column. They're fertilized inside the sponge, and then an actual larva is released from the female sheep's wool sponge. And on the right is a male sponge. These are spermatic cysts, and inside the spermatic cysts, those little purple points are actually individual spermatozoa that are released into the water column. So because histology expensive and we really didn't know what time of year the sponges were reproducing, we sampled adult donor sponges from May 2022 to April 2023 every single month except for October because we couldn't get out due to the weather conditions, in order to determine when they were most reproductive. And that way in year 2, we knew the times they were reproductive, that would be the time to go out and sample again gametogenesis in the propagules. So if you look at this figure, you can see that April and May, the sheep's wool reproduction for the adult donor sponges was the highest. So for year 2, we collected samples from propagules in the FW nurseries during the spring, since that's when we found the highest rates of gametogenesis. And here's an example of some of the sponges that we collected from. We collected from 6 different species. So on the top left is sheep's wool. In the middle is mountain glove sponge, grass sponge, another sheep's wool, and a yellow sponge. And so the good news is that overall, 88% of these 4 spongiid species were in gametogenesis. And that's a sample size of 95 individuals that I sampled. And then treated and stained using histology and used light microscopy to look for either oogenesis or spermatogenesis. And actually, not only were the propagules in the nursery, they were more fecund and they had a higher percentage of gametogenesis. So this is really good news from a sponge restoration policy standpoint to know that these animals are reproducing following restoration aquaculture. And so if you would like to learn more about sponge restoration, there is the Florida Master Naturalist Marine Habitat Restoration Special Topic. The different modules are seagrass restoration, which was written by Dr. Savannah Berry and Dr. Laura Reynolds. Coral reef restoration by Dr. Josh Patterson. Myself, I wrote the sponge habitat restoration module, and then there's another one on artificial reefs with Scott Jackson. And I would like to acknowledge EPA Region 4 Water Division, the South Florida program, for funding this research, my committee members and people who have helped me greatly, Drs. Josh Patterson, Ashley Smith, Shirley Pomponi, Chris Diaz, the team who's gone out in the field with me and performed all the histology, Harbor Branch Oceanographic Institute, the UF IFAS Tropical Research and Education Center, and the College of the Florida Keys. And all of this research was performed under the Florida Keys National Marine Sanctuary permits. So I suspect I'm ahead of schedule, but that does conclude what I have for my presentation.
All right, let's give a round of applause to Shelley. Thank you so much. So we will take questions from the room as well as from YouTube, and we will start in the room. And we don't have Dr. Mark Clark here as our ringer, so I'm going to rely on you guys to have a question for Shelley. Mary, for a moment. Yeah, go ahead.
Hi, I was wondering, what do you hope the broader implications of your findings with all these research in the nurseries and on sponges will be outside of maybe just your nurseries and the Keys?
So the nurseries as such are an area for them to cure and heal and then be moved into die-off areas. That's why we felt the reproduction study was so important, because like corals, if the animals aren't eventually self-sustaining, you just have to keep restoring them yourselves, and that becomes expensive and kind of counterproductive. So knowing that the propagules were so had such a high rate of gametogenesis following propagation, that's really good news that once these are moved to die-off areas, that they will recruit naturally. And we've already seen that in the nurseries. We've seen that it took about 8 years, but well, in the case of ours, but ours was a massive die-off area in the lower Keys, and it has returned to seagrasses and corals and just like a whole manna shrimp and sea urchins. I mean, every time you look under anything, there's a sea cucumber or a giant brittle star scurrying away. So to bring these ecosystems back to the former seagrass and sponge dominated systems that they were, because like in the case of stochastic events like hurricanes or things like that that are intermittent, once the hurricane comes and goes, that removed. Problems are in the areas where you persistently have low dissolved oxygen, you persistently have harmful algal blooms, and that's when it makes it hard to restore an area, because the cause from the beginning hasn't been removed. So we try to pick areas that have more of a long-term possibility of returning to the more of a steady state of seagrasses and sponges.
Awesome, thank you.
Thanks. YouTube is pretty quiet, so another question from the room? Yeah, please.
I had a question. We were just talking about how you want sponges to be the new oysters. In terms of their benefits, do they have any breakwater benefits in terms of like calming wave action or something like oyster reefs provide?
Yeah, just like seagrasses and oyster reefs, there's been a lot more studies into how oysters protect the upland and serve as breakwaters. But yeah, they do help to baffle the wind and wave energy. Like seagrasses, even though they're only about a foot off the benthos, they do help retain sediments and protect the upland. So limited studies on that, but they do seem to help baffle and protect the upland.
Yeah, sure. They have you hear a lot about oysters, a lot about seagrass. It's sort of like fun to snorkel in corals and what's your plan to give the sponges the makeover that they need to get, the environmental, it's easy to love a whale or a parrotfish or something, but how about a sponge? What's the plan?
I think sponges are very charismatic. And we are actually writing a field guide for sponge identification for shallow water sponges. Because as mentioned, the majority of the attention and even the research has focused on coral reef associated sponges. And some of these nearshore sponges, they're literally in like 2-3 feet of water and they don't have scientific names or common names. Sponges are actually notoriously difficult to identify. So working on a really intensive sponge field guide that will be published and printed this year. And the sponge restoration is growing. There's been efforts off of Pasco County. Biscayne Bay has started sponge restoration. We did get funded for the next 3 years to perform more sponge restoration in Biscayne National Park. And so people are really starting to appreciate the benefits of sponges in a similar way to oysters. I remember when I started my master's thesis back in, 2007, people were like, oysters, why are you restoring those? What's the benefits of oysters? And now, I mean, you can ask just about anyone and they appreciate how important they are for the environment, economy, protecting, property. And so I hope the same thing will happen with sponges in the future.
Other questions from the class?
Yeah, I believe I have one. So, bonefish are kind of near and dear to my heart. So when you mentioned like bonefish having like, do they have like a symbiotic relationship with sponges? Like kind of one coexists with the other, kind of like what kind of like mutually beneficial, like like I guess would say like how do bonefish like benefit from the existence of sponges? I was just curious about that.
Yeah, good question. So if you have nowhere to hide, it's a lot easier to become dinner. So one part about areas with a lot of sponges is that there's just more of that three-dimensional space, hide and forage. Also, they're really important at improving water clarity, water quality. They're removing a lot of the dissolved organic carbon and nitrogen out of the water. So in the case of bonefish, it's really the habitat quality and the ability to hide from predators. And also, there's definitely some forage. In the case of the juvenile stone crab, spiny lobster, once again, it's a place to hide during the day. And then at night to go out and forage. And if you've ever cut a sponge open, I mean, it is just filled with brittle stars and manna shrimp and crabs and snails. And I mean, I forget. I just read a study where it's actually the giant barrel sponge, but they pulled out like 61 invertebrate species out of a single giant barrel sponge. So, protection from predators, a place to forage, and a place to hide.
Thank you.
What would you say is the importance of someone who, for anyone who doesn't know about the ocean or what's happening in the ocean, why is this topic important for them to be caring about?
Well, healthy ecosystems equals healthy humans. Not only the drugs that sponges provide, the water quality benefits, the improvements to recreation, all of the seafood that we eat, seagrasses and sponge habitat are nursery grounds for more than 80% of commercially and recreationally important species. So if you don't have the nursery to rear and raise the juveniles, there will be a deficit in seafood for people to eat. So really healthy humans, healthy oceans.
Thanks. Thanks, Shelley. So what's your, when can we watch your defense?
Hopefully spring of 2026.
Okay, yeah, it seems like you're really far along, so congratulations on the work so far and good luck wrapping it up. And thanks again for for logging in to talk with us this semester. Maybe when you're finished, you can come back and visit us in person.
That would be terrific. I look forward to that.
All right, thank you very much. So for everyone in class, I want to check really quick who is our speaker for next week. You guys can always check on the website. Next week we have Vivek Sharma, and so he'll be talking about nitrogen and water transport in a rotational production system. So did we lose her? Okay, that's fine. So we're gonna go from sponges to cows, right? So we're gonna jump, and the water connection, of course, is that water that rains on a cow pasture that goes through the poop that takes the nitrogen down into the aquifer that goes to the springs that eventually feed the rivers that go to the coast. It's all connected. And I'm pretty sure Dr. Sharma is going to be with us here in the room because he's just up in live over, I believe. So, all right. So thanks everyone. We will see you next week, and everyone on YouTube.